All right, why don't we get started? Welcome everyone again to the third annual Species 360 Conservation Science Alliance Research Symposium. We have over 560 people from 67 countries around the world registered for today's exciting event. Again, I'm Jim Ginter, Species 360 CEO, and I will be your host for today's symposium. While we wait for, let's just go ahead and get started. Let's jump to the agenda. Here's a brief look at the agenda for today's symposium. Each presentation will be followed by 5 minutes of Q&A time. So we encourage attendees to please put any questions they have for the presenters in the Q&A function available in the toolbar below in your Zoom screen. And if you don't see it there, look under the three dots circled, labeled More, and you'll find it there. We have 6 intriguing and diverse talks today. We start in Australia, following echidnas after treatment and release to understand the outcomes of rehabilitation. Then a ZIMS comparison of 2 rarely studied gecko species, looking at breeding, timing, and mortality. Next, squirrels, and the hard question of which species earn a spot in zoological institutions. Followed by whether conservation priorities predict changes in zoo species holdings. After that, whether keeping carnivores far from their native latitude impacts their life events. And we finish with elephants and a rethink of how we measure survival in zoos. Every one of these projects is based on shared animal records, celebrating our community's collaboration through shared data. That's the key point for today. Before we get into the day's presentations, I want to take a moment to set the context. For those who may be less familiar, Species 360 is a nonprofit organization, and I'm pleased to share that you are the first group to hear our new mission statement finalized just this week. We create globally unique insights so wildlife can thrive. Through our platform, the Zoological Information Management System, or ZIMS, our members created the world's largest knowledge base on animals in human care. Over more than 50 years, our members have added millions of data points into ZIMS. Our mission has always been to make sure that data is useful for our members' day-to-day work and that it helps to further animal care and conservation. But we want to go further than that. We want to take that aggregated data and turn it into unique insights for wildlife. Insights that can further scientific research, animal care, and species conservation that simply wouldn't be possible without this global network and their amazing data. This research symposium today is a direct example of that mission in action. Every presentation you'll see today showcases how researchers are leveraging ZIMS data to advance our understanding of wildlife. And we hope to inspire others to do the same, to leverage aggregated ZIMS data so wildlife can thrive. Here's a short video which explains in more detail the role of data in ZIMS.
What does the weight of a tree kangaroo in Papua New Guinea have in common with the blood work of a red panda in Japan, or the water quality of a coral tank in California? On their own, they're important for the care and welfare of that animal. But when they join millions of other data points in the Species 360 Zoological Information Management System, ZIMS, they become part of a living, growing picture of the animal kingdom. Species 360 is a nonprofit organization that brings together more than 1,400 zoos, aquariums, and wildlife institutions globally. For over 50 years, our members have shared records in ZIMS, creating the world's largest collection of wildlife data. From daily animal care to managing entire aquarium ecosystems to informing strategies for conservation projects, each data point helps experts care for and protect wildlife. ZIMS helps ensure healthy, genetically diverse wildlife populations, while veterinarians use ZIMS to share and compare crucial medical data. When aggregated and anonymized, ZIMS data creates one of the most powerful resources for wildlife research on the planet, making a global impact on wildlife care, conservation, and research. It informs species reintroductions, helps combat illegal wildlife trade, and supports international wildlife policies and authorities like the IUCN Red List and CITES. Every record, every observation, every success story, it all starts with a single data point.
The Species 360 Conservation Science Alliance, or CSA, was formed to create wildlife insights. The CSA centers around the idea of a collaborative alliance of professionals working together towards a shared goal of species care and conservation. Our members record information about the animals in their care every day, And when that information is brought together, it becomes a global resource that no single institution could build alone. The purpose of the CSA is to put that incredible resource to work. It aims to maximize the use of ZIMS data to advance zoological conservation and academic research, and to support Species 360's mission of creating globally unique insights so wildlife can thrive. And this is why we hold this symposium every year, to showcase that it is what is possible when our community leverages shared wildlife data in research. I'd like to take a moment to thank the organizations that helped to make the work of the CSA possible. Thank you to the organizations listed here for supporting the CSA, helping to further wildlife and zoo research. If you'd like to support the CSA and help advance critical zoo and aquarium research, please scan the code in this slide to learn more. Sponsorship is what allows this work to continue and to grow. It supports the team behind the CSA, our work to fulfill research requests and related data analysis, and the collaboration that turns shared data into published research and practical guidance. Before we bring up our first presenter, I want to briefly mention that there are two ways to access ZIMS data for research: an Insights subscription and our research request process. The Insights program is an annual subscription providing access to Species 360's global resources in ZIMS, which includes access to our global medical resources, including anesthesia summaries, drug usage interactions, and expected test results, as well as global demographic resources in ZIMS, such as taxon profiles, age distribution, and pedigree explorer, among others. You can scan the QR code here to learn more. Research requests are for data that is unavailable or difficult to access directly through ZIMS. These requests are reviewed by our research committee and approved by our board of trustees. Our conservation and science team then extracts the data and provides support to researchers to help them interpret the complexities of the data. Before I introduce the first speaker, I'd like to quickly introduce some of our team who will be handling the Q&A following each talk. Florian Plard, population ecologist in statistics and modeling; Regé Nielsen, assistant research scientist; and Nanette Driver-Ruiz, senior product owner. Please remember to place your questions for the speakers in the Q&A box at the bottom of your screen. If you don't see the Q&A options at the bottom of your Zoom screen, look for the three dots in a circle labeled More. And with that, let's kick off the main part of today's symposium, the presentations. I'm pleased to introduce our first speaker, Dr. Adrienne Lavinia, Veterinary Director of Ballarat Wildlife Hospital and an adjunct senior industry research specialist at Federation University. Her work links clinical care with conservation medicine and post-release monitoring for native wildlife, and her PhD research on the health and ecology of Australian species leads directly in today's talk on the short-beaked echidnas treatment at Ballarat, demonstrating how clinical data, combined with telemetry, can inform disease status, survival, and habitat use without the physiological stress, financial cost, or logistical constraints of targeted captured studies. Thank you, Dr. Adrienne. Off to you.
Thanks very much for the opportunity to speak today, and thanks very much to the Species 360 team for organising this fantastic event. I'd like to share some of our research into integrating hospital and admission records with post-release monitoring of short-beaked echidnas presented to the Ballarat Wildlife Hospital. So a little bit of a background on the project. Wildlife hospitals play a critical role in the treatment and rehabilitation of injured and diseased wildlife, and they contribute to the understanding of biology, ecology, and conservation. But despite this investment, the short, medium, and long-term outcomes of clinical intervention still remains poorly understood. And we know that approximately 10,000 native animals are rescued, treated, and rehabilitated in Victoria each year, and that wildlife hospital records do represent a readily available but globally underutilised resource for wildlife health and conservation research. Ballarat Wildlife Hospital is a regional wildlife hospital servicing Central West Victoria, and echidnas are a commonly presented species. But we have limited baseline data on the health and disease ecology of this population, and there are no hospital post-release monitoring studies yet undertaken in this species. A little bit about the hospital catchment. Our hospital is in southern Australia, and it covers 9 First Nations lands, including the Wadawurrung peoples, which I'd like to acknowledge today. That's where our hospital is located. And it spans approximately 160 kilometres east, west, north and south. So that's about 5 million acres. So in our terms, that's quite a large catchment. And it features regional towns, farming, private lands, and a mix of state and national forest. So quite a mix of different landscape. Mongark, or short-beaked echidnas, are egg-laying mammals in the order monotremes, and they have a wide distribution right across Australia. So the aims of our research were to identify the primary causes of admission to the hospital, assess their baseline health with some pathogen surveillance, evaluate the influence of treatment and hospitalisation on rehabilitation and release outcomes, and then examine their landscape and understand the critical resources. So the methods that we used were the hospital admission records from Species360, clinical data records, necropsy reports, pathogen surveillance, and post-release monitoring. We integrated all these results and undertook some statistical analysis, which we use to inform clinical decision-making for the wildlife hospitals, shelters and rehabilitators, but also to inform conservation management with state and local government and community organisations. And also the observational data that was captured, we utilised the value in that for clinical decision-making as well. But for the purpose of today's talk, I just want to focus on the hospital admission records using our Species360 data and our post-release monitoring. So if we look at a quick summary of our admission data, we had 40 echidnas present over 3 years, 5 male, 30 female, and 5 undetermined. The most common cause of admission was dog attack, followed by vehicle strike, and then fire, entanglement, and then wounds and displaced echidnas. Each echidna that comes into the hospital has an examination under anaesthesia, and a set of parameters are documented, as well as radiographs, ultrasound, blood tests, and fecal exams. So that data is also captured in their clinical records and Species360 data. Animals that are treated and identified as potential release candidates have their final exam with tracking devices placed. And once they're cleared for release, they enter into the research project. After this point, they become the responsibility of the researchers, and they're tracked for 2 weeks, and then daily, and then fortnightly thereafter. And then once the trackers fall off or release, they are released from the tracking program. So strong collaboration from both the Wildlife Hospital team and the research team ensures good animal welfare in both directions. The transmitters are attached to the quills during their final examination under anaesthesia, and they naturally detach during the molting cycle. They're attached with a modified suture and adhesive bandage and glue. When we're radio tracking echidnas in the field, we're using VHF. So we are going out into the field, picking up the signal and tracking them via homing or triangulation, which are the 2 standard methodologies for radio tracking. And then we manually take a GPS location. So results so far, if we go back to our hospital cases and look at their outcomes, we can see that the dog attacks have a good prognosis for release compared to our vehicle strike patients, and that entanglement and displacement also has a favourable prognosis as compared to wounds and fire. But if we look at our released echidnas, we can see that we have a release rate of about 65%. So if we move on to some data from our echidnas that we've tracked, we can see we tracked this echidna for 9 months. She moved approximately 11 kilometres. And we can see that the early stage of rescue to release, she showed exploratory behaviour, followed by a period of settled behaviour. Another echidna that we tracked, we can see that we tracked her for 8 months. She moved a distance using the Haversine method of approximately 17 kilometres. And we can also see that in the early period, about 2.5 months from release, she showed that exploratory behaviour. And then for the 6 months after that we tracked her, she showed more settled behaviour alongside that road corridor. Another echidna that we tracked for 3.5 months did not show any exploratory behaviour and showed more settled behaviour for the duration of the time that we tracked her, and had a much less distance travel, just under 3 kilometres. And if we apply a heat map to her tracking data, we can see that the area under use, which we can also use kernel density estimates to estimate, was just under 1 kilometre squared. So of the 19 echidnas that we did track post-release, we can summarise their data into their presenting problem or reason for rescue and place them into groups. There we can look at their time in care, how long they spent in hospital, their sex, their tracking duration, their distance from their release site in week 1 and week 2, and then their total distance travelled. And then we can run some statistics on this data. So first we looked at their presenting problems and their groupings, and we wanted to compare that with distance moved and looking at week 1, week 2 and total distance. We used 2 different modelling statistical models, and we found no significance. Next, we looked at time in care, and we compared that also with distance moved in week 1, week 2 and total distance. And we also found that there was no significance with the 2 models that we used. So what does this tell us? Well, it tells us that there wasn't any detectable effect of presenting problem or time in care on post-release movement. And we need to clarify that there are limitations with the small data set, and we need to be cautious with our statistical model, which is why we didn't compare sex, because we only had 1 male that we tracked compared to 18 females. The other thing that we benefit from by doing the radio tracking and manually going into the field is being able to do a visual assessment of the animal post-release. So this enables us to look at body condition and behaviour to ensure that the animal is showing normal behaviour, is in good body condition. We can see that all the limbs are healthy and that the echidna is foraging and utilising the landscape. We can also look at the landscape that the echidna is using and what kind of canopy cover it has and what features are an important aspect of the landscape that the echidna is utilising. So our key observations were that there was no mortality. We did have 2 returns, but both were re-released without incident, and all echidnas released reintegrated into their environments. That their movement across the landscape isn't random, they show exploratory and settled behaviour, and that roadside remnant vegetation is a consistent feature. We're careful about making statements about home range because we don't actually know where the echidnas, whether they were in their home range when they were rescued. So we prefer to use area under use. But we do know that this home range or area under use does appear to be in what we previously considered were roadside corridors because they're areas where there are lots of remnant vegetation. And this has increased road crossing behaviour and risk of trauma. So we need to think about the release sites, and we need to think about whether those release sites have remnant forest And then the next part of the research would be to track the echidnas for a longer duration of time so that we can understand if there is actually a buffer size that's important in their release. The impact that our research has had has enabled survivorship data to be given to the rescuers and the carers, which has provided positive reception from them and the work that they do. It's also enabled our students to have placement opportunities. And by far, the greatest impact we've had is with the local residents surrounding those species release sites. So in summary, the study provides some baseline ecological data from an understudied population, and that we have some confidence that our veterinary treatment and our duration of care don't adversely influence post-release movement, and that post-release monitoring is a valuable tool to inform fine-scale conservation action. And that wildlife hospitals are not just centres for treatment, they're centres for disease surveillance, ecological research and evidence-based conservation. There's always lots of people to thank for these kinds of research projects, but I'd like to specifically thank the rescuers and all the students for their many hours of tracking in the field in all weather conditions. And I'd just like to say Nayatni Wiring Wiring, which is Wadawurrung, for thank you for listening with both ears. And does anyone have any questions? Thank you.
Dr. Lavinia, thank you. That's a wonderful update on this amazing species. And I must say, well delivered in the middle of the night in your time zone. Well done. I'd like to now open the floor for questions. If you haven't already done so, please place your questions in the Q&A box at the bottom of the screen. Nanette, do we have any questions?
Yes, I have one question to start you off with. You mentioned pathogen surveillance as part of the framework. What pathogens are you watching, and is there any concern about disease transmission between the hospital cohort and wild populations post-release?
So the clinical part of the clinical workup, we do fecal exams. And that's fairly standard. Also, ticks. So we have sent ticks off to a research facility. They've been looking at the diseases of the ticks as well. And at the moment, we're also undertaking some herpes virus testing in the population. So that's part of the passive disease surveillance. That's not part of our clinical workup. That all of that we'll put together as well as part of the study when all of that's complete.
Right. The next question: why do you think the pool is skewed so female?
Yeah, that's a great question. And I actually, I have thought a lot about that. And I do wonder because in other studies, in other movement ecology studies, the males have a greater home range than females. So you would think that it would be more likely that we would see males because they would be the ones that would be, showing the road crossing behaviour or expanding their territory. So unfortunately, as frustrating as I don't have an answer for that yet, but I do hope we can come up with some potential reasons or the data yields some information that we haven't yet elicited.
What are the impacts and benefits of these studies in zoological systems?
I think that understanding how echidna health is managed in care, so whether that's treatment for diseases or just management of general health. So a lot of our entangled and displaced echidnas aren't necessarily diseased. They are just emaciated, anorexic. So they require more rehabilitation than they do hospitalisation. And echidnas are very tricky and challenging to keep in captivity, especially wild ones, because although they're quite small, they're very strong, and they can be prone to self-trauma. So their enclosures in zoo facilities need to be large enough that they can exhibit normal behaviour without self-trauma. And they are prone to stress with new introductions. So when you're introducing a new echidna to a collection, you need to be very careful about stress. That we can have coccidia outbreaks in collections when a new echidna is introduced. So understanding management of wild echidnas can really help inform care in zoo collections as well.
Did any of the echidna that you were studying die during monitoring post-release, and what was the cause?
No, I'm very happy to say we had no mortalities. We did have 2 returns, and just for time, I didn't elaborate on those. One echidna we released, and this was a peri-urban echidna, so very, very close into an urban area. And we released it in a small wetland, roughly in the centre of this small regional city of Ballarat. And about 24 hours after we released it, it was found in the car park of a car dealership. So we literally picked it up, checked it over, and then re-released it in a slightly different place. And then after that, it didn't end up, displaced in suburbia again. So that one was a successful reintroduction. And the other echidna we had in was a bushfire echidna that had come in and was in care, I think, for about a week. Its blood test results and its X-rays were clear. We released it, and then about 3 days after tracking it, one of the team was a little bit concerned that it was showing some abnormal behaviour, so it was re-rescued and brought back in. And then when we reran its blood and X-rays, we could see that actually some changes had begun to occur, and it had some smoke inhalation, so some congestion to the lungs, which was progressing towards pneumonia. So it spent longer time in care and then was re-released and reintegrated well, and didn't require re-rescuing again.
Thank you both. That was very insightful. Our next speaker is Sam Parsons, a researcher with Bristol Zoological Society, whose work focuses on the ex situ management and scientific understanding of underrepresented reptiles and amphibians. Today's talk involves 2 understudied geckos, the Henkel's leaf-tailed gecko and the turquoise dwarf gecko, and asks what ZIMS data can reveal about their demographics in human care, and how those insights might strengthen breeding programs for species still lacking published guidance. Sam, I'll turn it over to you.
So, hi, I'm Sam. I'm a postgraduate of Zoological Management and Conservation at Bristol Zoo Project. And I'm going to talk a little bit about a study involving captive demographics for a pair of underrepresented gecko species, these being the turquoise dwarf gecko and the Henkel's leaf-tailed gecko, left and right. So both of these species are represented across EAZA collections, EAZA being the European Association of Zoos and Aquaria. These are also both species that we have here at Bristol Zoo Project. And both species are very underrepresented. There's not a lot of literature available for either. However, the Henkel's leaf-tailed gecko has best practice guidelines. Both species are managed at a population level for breeding programs or for stud books. However, only the Henkel's leaf-tailed gecko has best practice guidelines. This is incredibly important for just ensuring that husbandry methodology is sort of ubiquitous across collections and helping to just guide the species in the right direction for conservation. So the lack of this for the turquoise dwarf gecko does mean that there are gaps in the literature that should be addressed, and the turquoise dwarf gecko is currently classified under a greater level of threat than the Henkel's leaf-tailed gecko. So I'll talk a little bit about the species. This is the Henkel's leaf-tailed gecko. It's a Malagasy endemic species found only in these regions of Madagascar. Its habitat is fragmented. It is threatened by illegal logging. It's harvested for the pet trade, both legally and illegally. And it is incredibly difficult to study. It is very difficult to find in the wild. It's capable of some extreme crypsis, as you can see in the bottom right here, which means that it's really difficult to measure the extent to which it is threatened. It's difficult to learn about its ecology and its behavior. And it's very difficult to quantify any kind of population metric. There is no estimate for population, and the last IUCN evaluation was in 2010, which is very old and long overdue for a sort of subsequent evaluation. Similarly, the turquoise dwarf gecko is a Tanzanian microendemic meaning it inhabits an incredibly limited range, in this case 8 kilometers squared. It is only found in this tiny orange region in Tanzania, the Kimboza Forest Reserve. It's similarly threatened by harvesting for the illegal pet trade. However, the reserve is also permeated by invasive plant species, and the turquoise dwarf gecko is entirely dependent on a single species of screw pine, this being Pandanus rabaensis, this tree in the top right here. And so this can have some really significant implications for the species at a population level if its only viable habitat is being driven out, right? So this species live in social groups, which is unusual for a reptile. They live in male-oriented polygynous social systems where one male will monopolize a group of females and his offspring, and they'll normally live on a single leaf or a single trunk of these trees. The species is sexually dimorphic. Males are this brilliant blue color, as you can see here. Females are a less brilliant, but still very pretty, coppery, browny color. And as such, this species is much easier to locate. It's quite conspicuous. However, it still is understudied. The last IUCN evaluation was in 2012, and it still doesn't have any accurate population estimates. And much of its behavior and ecology is unknown. Particularly due to its sociality. It's unclear. Wild sex ratios have not been studied, and things like its seasonality are somewhat understood, however, very, very seldom comprehensively studied. So our study aims to compare longevity, population growth, and breeding rates between species. This is super important for captive population management. If we are attempting to conserve these species, we're attempting to maintain a captive reserve population, then we need to be able to maintain them in captivity at a sort of healthy demographic level, right? Also, descending captive sex ratios is super important for captive management and for informing just wild ecology. For a species as understudied as the turquoise dwarf gecko, for example, that also exhibits very complex male and female relationships in the wild, understanding these in captivity can help to inform husbandry methodology, and we can learn about the wild from these individuals. Similarly, the Europlatus henkeli, the Henkel's leaf-toe gecko, is studied, has been subject to more study. It's got best practice guidelines. So how do these best practice guidelines influence how the species is kept in captivity? How does it help us to care for the population? And can we use this to inform the turquoise dwarf gecko and the sort of captive methodology that we employ there? Also, we evaluate seasonality and biologically significant milestones for Lygodactylus williamsi, the turquoise dwarf gecko. These have not been comprehensively studied in captivity before, or just ever before, and they have some quite significant implications for captive management and conservation in the wild. This is a massive slide, and I don't have time to talk about it comprehensively. So what we essentially did was obtain taxon reports for both species in captivity from April of 2021 to April of 2026. These records are not the entire captive population in Europe. It is just records who were changed during this time. So if an individual was born in this time frame, or if they died in this time frame, then their records are counted. Individuals who didn't do anything in this time were not counted. Similarly, we filtered out grouped individuals, ZIMS listings with enclosures with multiple individuals, as these records sadly don't have information on the exact date of birth or death of individuals, because of course it's a large enclosure with multiple individuals, so that's just not possible. And we also manually filtered out institutions that were not EASA affiliated or in Europe. This is so that we can get a more clear picture of seasonality ex situ to see how this might affect the species. So we are interested in looking at population growth, mortality rates, lifespans, sex ratios, seasonal breeding patterns for the turquoise dwarf gecko, and mortalities per birth month. Most of these were analyzed descriptively. However, lifespan, we used man-Whitney U-test. Sex ratios, we used a binomial test. And for seasonal breeding patterns, we used chi-squared. So these are the raw numbers we observed. There were 180 records for U. henkeli, the Henkel's leaf-tailed gecko, and 608 for the turquoise dwarf gecko. So immediately we're seeing that there are there's a lot more sort of movement and changes in the captive turquoise dwarf gecko population. I believe, unfortunately, or fortunately, depending on how you look at it, a large number of Henkel's leaf-tailed geckos were moved to the United States, which means we can't study them, but also I'm sure they're going to live their best life over there. So it's worth mentioning that the Europlatus henkeli population in captivity in Europe was significantly smaller than Lygodactylus williamsi at the time of study. So for population growth, we observed a very healthy trend for both species. The turquoise dwarf gecko, the rate of population growth, this being simply the number of deaths subtracted from the number of births, actually doubled over the 5-year period, which is really great to see for such an understudied species. It shows that the species is capable of self-regulation in captivity, and that something we're doing is working in captivity to help us protect the species in the event of wild collapse. Similarly, Europlatus henkeli did fluctuate a little bit more. However, the population was smaller and a lot of individuals were moved to the United States. But we do see a general upward trend, which also does indicate that the captive population in Europe is capable of sort of self-regulation, and that we have a sort of stable stock for captivity. For biologically significant milestones, we identified the first 30 days as a really significant place for both species. Within these first 30 days was the highest number of infant and juvenile mortalities. However, Europlatus henkeli tends to stabilize much faster, whereas turquoise dwarf gecko had a much more sort of volatile stage of life within the first 300 days to a year. This generally matches what we know about the species ecology. Europlatus henkeli, the Henkel's leaf-tailed gecko, is solitary. Mating events will be quite transient. They don't have complex social systems, and it's a species who occupy quite a broad range. And so we expect them to be comparatively more R-selected than the very social, very densely populated Lygodactylus williamsi. So this matched our hypotheses generally. Captive sex ratios was something that did not match our hypotheses at all. Both species appeared to be male-oriented in captivity to varying levels. I'd like to draw your attention to this p-value highlighted in red. A high number here is associated with a more even ratio, and a lower value is a less balanced ratio. So we see a p-value of 0.678 for Europlatus henkeli, meaning that sex ratios are generally quite balanced in captivity. This wasn't particularly unexpected. We weren't expecting one sex to have a sort of particular weight over the other. However, for turquoise dwarf gecko, who is polygynous and live in harem groups in the wild, we certainly expected there to be more females. So for a p-value of 0.057, this shows that sex ratios are incredibly imbalanced. However, unexpectedly, they were unbalanced towards males. And for every one female, there are 1.34 males, which doesn't match what we know about their ecology at all. So there are a couple of explanations for this. One is temperature sex determinism. Both species, as reptiles, the temperature at which eggs are incubated influences the sex of the individual. And so it's possible that some error or aberration in our captive methodology is leading to a disproportionate amount of males being born in captivity. It's also possible that there's some causal mortality for young individuals. Immature individuals are very, very difficult to reliably sex. And so if there is some hitherto unknown cause killing off a lot of individuals of a certain sex whilst they are below these biologically significant milestones, then that's also another possibility. And finally, it's also possible that wild ecology just isn't understood enough. Like I said, this species lives in polygynous harem groups where a male will monopolize multiple females per colony. However, there are some studies which have observed multiple males on single trees. It's possible that they observe that they perform sort of bachelor groups, similar to what we would observe in mammals with similar social systems. However, this is all very understudied and not widely known. So similarly, the species does appear to be a seasonal breeder. Births persisted year-round, however, they were definitely concentrated in certain months. We generally observe more births in seasons we'd consider naturalistic. Naturalistic means a few things in this case, the first of which being the natural breeding season. In the wild, they'll breed in the Tanzanian rainy season, which occurs in November to February, March. And we do observe a spike here beginning in November with an enormous amount of births in January. It's worth mentioning that this disproportionately high number of births in January will likely be manipulated by institutional sort of practice. It's common practice that if an individual's date of birth is unknown, it's listed as the 1st of January that year, so you know what year it was born. But even despite this, there were an enormous number of births in January, which matches these species' naturalistic breeding seasons. Similarly, we observe spikes in April to May and from August to September, which matches with European seasonal shifts. So in the wild, things like increased humidity, increased rain, or increased temperature may induce breeding behaviors. And this is the exact time of year here in Europe which we would observe those kind of seasonal shifts. And so it's also entirely possible that that's what's happening here. This chi-squared value of 0.001 illustrates just how unbalanced this ratio is. A higher value is associated with more even distribution. And this is, of course, an incredibly uneven distribution across the months. We also observed differences in longevity per birth month for Lagodactylus williamsi. This is attributable to a few things, none of which are known for certain. One explanation is this is some form of seasonality. It's been observed in some South American species of Lagodactylus geckos that the female reproductive organs will actually grow in size during breeding seasons. So there is a biological link between gecko and season. In some species, this has never been observed in Lagodactylus williamsi, but it's a possibility. Another one is just aberrant husbandry. This species is incredibly sensitive to temperature changes, to its sort of changes in environmental conditions. And so months like November, where we observe a significant drop in longevity, could be attributable to seasonal temperatures getting colder, more individuals die because of life support systems failing, or simply just the conditions changing too fast for the geckos to adapt to. So overall, the study definitely yielded some pretty interesting results. I would like to acknowledge a few limitations first, the first of which being the small data pool for the Henkel-Leafto gecko. There were not as many individuals as we would have liked to study. However, we still had quite a healthy data pool. Similarly, there are many records in ZIMS which, through no fault of institutions, were difficult to provide data for, these being sort of grouped enclosures where entire sort of colonies of individuals are classed in a single record. So these records will likely represent very valuable breeding colonies. And so not having these records is unfortunate for painting a sort of holistic picture of the entire captive population in Europe. However, the study was successful in comprehensively studying demographics of the turquoise dwarf gecko in captivity. This is not something which has been done before, so this is definitely an important first step. And similarly, there are some definite avenues for future research of these species which have been illuminated thanks to the study. So that's all from me. Thank you so much for your time. I have some time for questions now, but my email's here if we don't have time for yours or if there's something you want to contact me about. So yeah, thank you very much.
Brilliant insight, Sam. Thanks so much. It's always encouraging to see the growth in knowledge of understudied species. Rege, we'll have to be a little bit mindful of time, but can you please guide us through the questions for Sam?
Yeah, absolutely. One of them is if you know if there are any plans of developing best practice guidelines for the dwarf gecko.
I do not know of any currently.
No. How old do these two species have to be able to reliably sex them?
I'm not actually sure. I believe it's probably around sexual maturity, sometimes a bit before in some gecko species. I'd assume for the turquoise dwarf gecko, it'd be around the year mark when the individuals start to become fully mature. The Henkel's Leafto gecko, it might be, I would assume before, but I do not know for certain.
Mm-hmm. And then you mentioned that you only use the individual records and not the group records. Do you know the kind of the percentage or the presence of group records and how that would have affected your research?
So my study focuses on how the demographics of the captive populations are changing. And so things like grouped sort of colonies or sort of enclosures with multiple individuals will have some really, really valuable breeding sort of happening in them. And so losing out on these is a great shame, particularly because these will be, in many cases, the most productive enclosures for creating new individuals and for breeding. And so, yeah, it's a shame to not have these sort of the birth data. However, generally, a lot of individuals think things like births and deaths, we still can get a picture of what it's like across the population, despite not having these.
So the multi-dollar question is, what will be your advice for the record keepers?
Yeah, just keep doing what you're doing. It's of course expecting people to keep records of these enormous enclosures with multiple individuals housed in them. It's not reasonable, right? It's not possible for human beings to do, let alone already overworked zookeepers, right? So the records we have are excellent. Yeah, it's the biggest drawback was the lack of Henkel's leaf-toed gecko data. So if we had access to if we just used global records across institutions worldwide, that would have been really valuable as well. And that would have mitigated a lot of the sort of desired data which we didn't get. But maybe some future study could do this. It's difficult with this study due to seasonality in Europe being quite fixed, whereas getting a global study would mess with that a little bit. But yeah, no, the records are great. Keep doing it. It's good.
Thank you. Yeah, so the individual data for sure. And I think this is a nice example of how ZIMS data for species that have been kept for years but studied very little can be really helpful for the demography. Thank you, Sam.
Thank you.
Yeah. Thank you, Sam. Thank you, Reggae. Yeah, it's a great reminder for those of you that care for wildlife and human care, especially if you have small populations or understudied species. Collecting that data is so incredibly valuable. So thank you for those that are doing it, and just encourage all of you to do more of it. So thank you. Our next speaker is Dr. Irena Schneiderová from Charles University in the Czech Republic. Dr. Irena is a researcher at Charles University in Prague and a bioacoustic specialist whose work spans species from Eurasian ground squirrels to bush babies, slow lorises, and hyraxes. Drawing on 3 years hands-on experience at Prague Zoo, her current research covers animal husbandry, the management of mammals in zoos, and reintroduction programs. Today's talk explores whether the world's zoos give this widespread but undersupported group the conservation attention it needs. Dr. Schneiderová, off to you.
Okay, so hello everybody. Today I want to share with you our research focusing on squirrels in zoo collections. This research was a part of a bachelor thesis of my student, Markéta Dušková, and we are doing this together with Gerrit Werenberg from the University of Oulu from Finland. So some introduction to squirrels. So squirrels are belonging to Sciuridae family, which is quite diverse, widely distributed, and I would also say quite popular rodent family. This is ecologically variable family. Also, we have the species that are diurnal or nocturnal here. We have a species that are arboreal or terrestrial here. And we also have a species that are rather solitary and also those that are forming more complex social groups. So for the zoos, the family can be also very interesting from the educational perspective. But despite the fact that the family is quite popular, so it seems that only little is invested in research and conservation, and more than 90% of species remain understudied. If we look at their conservation status, we can see that the majority of the species is evaluated as least concern. However, we have about 10% of species that are threatened. So it means they are either vulnerable, endangered, or critically endangered. And we have 3 critically endangered species here. It's, for example, this Vancouver Island marmot or this flying squirrel from India. So zoos can have really important role in conservation, animal conservation. And if we look at this one plan approach, focusing on integrated conservation, so we can see that the aim of the zoo is mostly the management of ex situ populations to meet the conservation goals. However, we know from some studies that zoos tend to keep rather charismatic and attractive species. And if we look at a particular taxa, we can see that they, because of the capacity limitations, they usually can keep only a small percentage of species, and even smaller percentage of these species is somehow threatened. So our aim was to look or check what is the situation for the squirrels in zoos. And we also wanted to see whether there are some conservation, in situ conservation projects or some reintroductions done with squirrels that are bred in the zoos. So for this, we had to compile the list of extant species. That was the first step. The second step was to use the zoological information management system to make a list of species that are kept, to get the number of holders, number of individuals for each species, and also for each species we received the birds within the last 12 months. We also made a comparable list of species that are kept from the website Zootierliste. And for each species, we also added the conservation status according to the IUCN Red List of threatened species. And finally, we made a literature review based on specific keywords to find whether there are some in situ conservation projects or whether there are some reintroductions done with the squirrels born in zoos. So here we can see the first results. So we can see that the zoos keep 82 species. We can also see, if we consider both databases, we can also see that the both databases differ a little bit. However, they are highly correlated. Finally, we can also see that if we look at the specific taxa within the family, we can see that the number of species kept corresponds to the number of recognized species. So simply said, the larger the group is in the wild, in the nature, so the higher the number of species that are kept in zoos for most of the taxa. Considering the conservation status, we can see that only 5 species kept are classified as threatened, and 76 species are not classified as threatened. So it's the majority. However, if we compare it statistically, it is not significantly different from threatened and non-threatened recognized species. Here we have the bar plot showing us the number of holders for the species. And we can see that the most frequently kept species is the black-tailed prairie dog. Maybe this is because it's a quite popular species for the visitors, and you can also see that the species is highly overrepresented in comparison to other species. The second most frequently kept species is the Prevost squirrel. Maybe this is because the species is somehow attractive visually. Here we have another bar plot. It is a little bit more depressive because it's showing the number of individuals kept according to ZIMS. And First, we can see that almost half of the species recorded in ZIMS are kept in less than 10 individuals. And we can also see that the black-tailed prairie dog is really highly overrepresented in comparison to other species. You can also see that the most frequently kept species are usually those that are evaluated as the least concern. The only exception is the European ground squirrel, which is endangered. Here we have another little bit pessimistic chart because it is showing you that almost 3/4 of squirrels that are kept did not breed within the last 12 months. So we can see that the most of the species are not actually breeding in zoos. But if we look at this chart, which is showing the species that are breeding in the zoo, we are receiving a little bit optimistic, little bit more optimistic picture, because we can see that those that are somehow threatened and are used for reintroduction projects, I will talk about it later. So these are relatively highly ranked. So the European ground squirrel has this first position, and also you can see that the Vancouver Island marmot is also relatively highly ranked. It's probably because this species is used in situ conservation project for introductions. And we can see here that even if the species is kept by only limited number of holders, in this case there were 2 holders in ZIMS, so they can produce a lot of juveniles if it's for reintroductions. So at this point, I would like to say that we have to be cautious when we are interpreting our data, because there might be some bias. The first thing is that not all the zoos contribute to ZIMS, so the number of holders and individuals might be underestimated. Second, data can be outdated or incomplete for small rodents living in large group, because it's more challenging to make the good record keeping for these small mammals. And also the data might be biased for colonially living ground squirrels or prairie dogs, because they are often kept in such semi-natural enclosures. And here it is also very hard to make a proper record keeping and make a proper population estimation. So to sum it up, we can say that less than 30% of sciurids are globally kept in zoos. This is less than for other taxa that were investigated in this way, but it is also more than for other taxa. However, more than half of these species kept, they are kept by or fewer than 5 institutions. The population numbers for nearly half of the species do not exceed 10 individuals. Only a quarter of the species reproduced within the observed 1-year period, and more than 90% of the species kept are not listed and as threatened. So it seems that limited effort is dedicated not only to research and conservation of sciurids, but also maybe to zoo collection planning and developing husbandry protocols. However, to be more positive, we can see and say that zoos played a significant role in conservation of sciurids, because at least for 2 species, there are very good conservation projects. It's for the Vancouver Island marmot and in Canada, in North America, and then it is for the European ground squirrel in Europe. Finally, we can also say that the zoos, they can engage in conservation not only of globally threatened species, but they can also focus on conservation of locally threatened species. So, for example, in Europe, this challenge could be accepted for the Eurasian flying squirrel in the future. So, thank you for your attention. Thank you, the organizers, for organizing this amazing event, and I am ready for the questions.
Thank you, Dr. Schneiderová. What an insightful talk about this amazing group of species. Reke, what questions do we have?
We have one question asking if you have considered how the regional assessments might affect the collection planning for the institutions, like the black-tailed prairie dog that are threatened in Canada.
Yeah, I can imagine. This is what I was talking about in the last slide, that for us sometimes it's hard to evaluate it globally because the species are not threatened globally, but can be threatened locally. So I can imagine this is happening for some of the species. And yeah, I know that there are also some reintroduction projects done with the black-tailed prairie dog. But yeah, that's true. I cannot say it's not. However, maybe I think if we evaluated only the Europe in the same way, I think we would get the same result. So I think that the black-tailed prairie dog, it might make sense in North America, for example, but maybe the situation should be different in Europe, for example. So I will answer like that.
That makes sense. And then you mentioned yourself that not all institutions are recording their data in ZIMS, and also that these are colonial species that are likely underestimated in zoos, both of the way that they are living and the way they are kept. Do you have any idea how or if the breeding picture will look very different with more complete data?
I don't know, but I just have my own experience because I am doing the control monitoring for some of the zoos. I am doing the control monitoring of the European ground squirrel, and yes, I know that they are living in these enclosures, and each year we are capturing several juveniles. So we report it to the zoo. They will make the note. Maybe they will record that in the ZIMS. But I'm sure that there must be more juveniles in the enclosure. Yes, so they are just recording what we tell them based on what we captured. But in this case, I'm sure it's underestimated.
Absolutely. Do you have any idea how big the problem is about the data quality, like if it's recorded in as individuals or in group, or if it's used as colonies, like recording it with census data?
I think I cannot say. Maybe this would be answered in much better way by somebody who is working at the zoo, who is having the practice experience. But I know if I go to some zoo and I see the colonies of the black-tailed prairie dog, for example, so I can see that they are kept in such enclosures where I can imagine that the record keeping is very challenging. And I'm sure that they don't have the exact numbers of these animals. So maybe this is the question that should be responded by somebody who has the hands-on experience.
Absolutely. The last question I have is if you know if the global zoo community have a policy concerning these tiny population that you found for many of these squirrels. Like, is it recommended that they are phased out, or that they are fewer, or that we keep fewer species but in bigger groups? Or is it really species dependent? What the recommendation will be with these small populations?
I'm not sure. This is also the question for somebody actually working at the zoo and doing this collection planning, but as far I know, I would say nobody cares maybe because yeah, I think I don't know any like a program apart from that European ground squirrel and the Vancouver Island marmot, which are somehow like dealing with the species in more detail. So I cannot say.
Thank you. I think your talk really shows us that squirrels are easy to put in a category as being common, but then you really showed that we might be costing us some conservation opportunities by doing such. Thank you, Irena.
Yes, thank you. Thank you for the wonderful presentation. And thanks, Ricky, for the questions. We'll move on. So our next speaker is Species360's very own Hannah Jenkins, who kindly agreed to step in at the last minute, literally yesterday, to replace a speaker who was unfortunately not able to present today. Hannah is the Species360 Conservation Data Lead. She has also just submitted her PhD thesis on the very topic she will be talking about today. Hannah, the floor is yours.
So yeah, I'm going to talk to you about some research that's just been accepted for publication, looking at whether conservation priorities predict changes in zoo species holdings. So first, a little bit of context. So zoos have four main pillars: conservation, education, recreation, and research. And modern zoos are increasingly positioning themselves as conservation centres, rightly so. But what we wanted to look at is whether the zoo collections themselves reflect this conservation priority, and specifically whether zoos are responding to changes in conservation status and whether they're allocating resources to species with higher conservation need. So to put that in a little bit of a visual context, if we have a frog at a zoo that is least concern and that frog becomes critically endangered, are we increasing the resources that we're putting into that species? So are we holding more of that species, and are more zoos holding that species? And conversely, if the species goes from threatened to non-threatened, are we moving those resources away from that species? So to do this, we did the first large-scale retrospective analysis of zoo collections, and we used ZIMS data to do this. So we looked at 31 years of zoo data from 1990 to 2021. We started in 1990 because this is when electronic record keeping became standard practice. So it was more, we were more confident that a whole collection would be added to the database, to Arks at the time and now ZIMS. The data set contained data from over 600 anonymized institutions, and we received data for over 7,000 tetrapod species. And with this data, we then looked at changes in the number of holders per species per year, following changes in those conservation criteria. And those criteria were identified in a previous paper as criteria that might affect species panning, and that's IUCN, CITES, and breeding programs. So for this study, we looked at AZA, EAZA, and WAZA. And to answer this question, we looked at holdings for each single species throughout the data set, and we looked at the number of holders over time. Then for each species, we looked at whether it had a change in that conservation criteria. So for example here, moving from non-threatened to threatened, and then whether the trend in holders increased over time, so had that positive trend, whether it decreased over time, or whether it stayed neutral. So it had a change of less than one holder per year. And so what we would expect to find from the data is as that conservation importance increases, the number of holders increase as we're putting our resources into that species. And as conservation importance decreases, that we're moving away from that species and less zoos are holding it. So what we actually found, starting with IUCN, so we looked at all of the linear regressions per species and grouped them all together into positive trends, neutral trends, and negative trends. And we looked at this for species that were moving from non-threatened to threatened at the top of the table and threatened to non-threatened at the bottom of the table. And we looked at all species together and then also taxonomic groups. And what we'd expect from IUCN is that those species moving to threatened would have positive trends as we're investing, and those moving to non-threatened negative trends as we're de-investing. But what we actually found were most of the species had neutral trends. This was the case across most taxonomies, apart from Mammalia, which had a 50-50 split. And we found that 76% of uplisted species showed neutral trends, and 80% of downlisted species showed those neutral trends. Only 19% of uplisted species showed positive trends, so only 19% had that increased resource in holders. And downlisted species, so species that were becoming non-threatened, did not decrease in holders. So overall, we found that IUCN threat status was not a strong driver of collection decision making. It did influence certain species, but holistically across the board, it wasn't a strong driver. For IUCN, we have, CITES, we have a similar story. Most of the trends were neutral, but we have a bit more spread across different taxonomy. But 69% of newly restricted species showed those neutral trends. Only 22% of species increased holders as restrictions increased. And responses were highly species specific, so there was a lot of variation depending on the species. And overall, we've determined that CITES was not a strong driver of species planning on its own, but CITES was also more difficult to interpret than IUCN because it does have many competing implications. So when a species becomes more restricted, it does mean that it could be more could have a higher conservation value, but it also makes it more difficult to move between zoos because there's a lot more red tape. Looking at breeding programs, this is where we saw the strongest positive trend. So no species that had very few species that had a breeding program introduced had negative trends. Most had neutral, but we also saw this strong correlation with the positive trends. And 36% of species increased in holders post-program introduction. So if a species had a breeding program introduced, 36% of them increased in those holders. And it was found to be the strongest predictor for an increase in holders across the 3 conservation criteria. What we also found, though, that some of the species with the strongest increases in number of holders per year were actually non-conservation value species. So these might be species that are least concern, but they have a breeding program set up to monitor the population, if they're a popular species, or if they have different breeding program roles that aren't direct conservation roles. So to sum that up, what we found was IUCN and CITES were not strong drivers of species planning decisions, but breeding programs were. And so that really highlights the role that breeding programs play in zoos, and that the importance it is to choose those species appropriately so that we can make sure that we're investing all those resources in the right place. So we attempted to look at why some of this was the case. And I say attempted because it's very complex. And that's basically how we can sum it up. Species planning is very complex. It's not just about how threatened a species is. It's not just looking at the individual species and saying, is it endangered? Is it controlled? Does it have a breeding program? A lot of the time, you're not just looking at that single species either. You're looking across the board at your entire collection and how that collection can then provide value for your different pillars. So provide education value or recreation value. We also know that zoos have competing pressures. Conservation is a huge role for zoos, and we do really great things. But zoos are also businesses, and they also need to manage their business profit. They need to generate funds so that they can contribute to conservation, so that they can increase visitor footfall, so they can improve welfare. They need to be successful and thrive. So just to kind of summarize, so it was a bit quick, but we found that zoos were not systematically responded to changes in conservation criteria, but that didn't mean that there wasn't a lack of action. We found that a lot of the time there was a very species-specific action. We could really see those species that people had rallied around and increase the holders. So when there was an event that happened, say something became assessed as more threatened, or there was an environmental impact to that species, that then there was an effort to bring it into ex situ conservation and then provide those resources. But it's very resource heavy. So it's not something that can be done for every species. And we see that reflected in the results. Species planning is very complex, and there's a lot of interplaying different pressures that can conflict with each other. But if we try to manage these pressures holistically, then we can improve that benefit to conservation overall. So I just want to say a quick thank you to all the Species360 members, because this research definitely wouldn't have been possible without everybody contributing their data. And then just a quick thank you to my supervisors and collaborators at the university as well.
Hannah, thank you so much for those insights on species planning. What a complicated decision process. Thanks for providing the information on how that's changing over time. And thank you again for stepping in at the last minute. Nanette, do we have any questions for Hannah?
Yes, we do. Since changes in IUCN designation occur at different times and CITES designations also occur depending on status in the wild, did you take into account the changes in holder according to the time frame? For example, two-toed sloths had a new designation since the last hop to CITES 2.
Yes, I think I understand the question. But yeah, so each species was treated individually. So the change, each change, so IUCN, CITES, and breeding program were all treated independently. So we had, if one species had multiple changes for 3 different criteria, they would have 3 different linear regressions. So they go into each different sample set. So the linear regressions were based on when those species changed for each criteria.
For species whose holdings are growing and not for conservation reasons, any idea of these are the species that are thriving the best in zoos in terms of health and well-being? So for these species, great welfare means ease of care and great exhibits for the public.
So we didn't look at this species in terms of welfare. We had to limit our scope because this is a project that could increase substantially. But what we did find from the species that kind of were at the top of the increase in holders, where they were a lot of popular species, sort of those known entities. I think meerkats was one of them. So it did seem to have, I think there were actually some domestic species up there as well. So there is definitely a visitor perspective coming through in those species planning decisions.
Did you see a difference in historic and more recent trends? In other words, are zoos now more reactive to IUCN uplisting or program species?
We didn't look at that. So we just looked across the board, but that's definitely an area that we could look at in the future to sort of segment it into 2 different time periods potentially. I think the hardest thing might be trying to work out when that cutoff is of what a modern zoo is, and also or when those changes came into play and more people are reacting that way. But also the lag of change is something that's really important to keep in mind. That's why we try to open it up a timeline as much as possible, because it does take zoos time to respond to these changes as well. It's not like something can be assessed next year and then everybody gets it into their organization.
Do you think zoos and aquariums should focus their breeding work more on endangered species, even if it is at the expense of their businesses?
That's a very political question that's difficult to answer. I think there is a balance. They're always going to compete against each other, but there is potentially a middle ground to sit in. But if you focus all of your energy onto endangered species, you're doing great things for conservation. But if you can't open your zoo doors and you're not getting that visitor footfall and generating those funds, are you going to have the resources you need to breed those animals? So I think it is a big balancing act, and lots of things that need to be evaluated against each other. It's not a simple question, a simple answer, unfortunately.
And I think this might be the last question. What do you think might contribute to mammals having such a high percentage of positive trends following a change from threatened to non-threatened?
I think mammals are very popular species, and we know that they're a big visitor draw. So I think even if they become less threatened, zoos might still want to invest in those species. I also think with some, especially the larger mammals, it's much more difficult to move that species on. So if you have a tank of amphibians and you just give the whole tank to another zoo, that's quite an easy transfer. Moving an entire herd of elephants to another zoo is very complex. So there's probably a lot more to consider with those mammal moves potentially.
Thank you, Hannah.
Very nice. Thank you. Thank you both. Our next speaker is Samantha Ward, Professor of Zoo Animal Welfare and Legislation at Nottingham Trent University, where she leads research that bridges academic science and zoo practice, from developing welfare assessment tools to advising major zoos and charities on ethical standards. Having begun her career as an animal keeper before moving into research and academia, she brings both hands-on experience and scientific insights to her work on evidence-based management and welfare. That focus underpins today's talk, which draws on decades of ZIMS data to ask how living outside their natural latitudes shapes the lives of zoo-housed carnivores. Dr. Ward, your floor is yours.
Thank you. Okay, so I'm going to be presenting some of the research that I've been working on in a collaboration with the co-authors, as you can see on the screen here. It's been quite some time in its making, but we've had a variety of different problems because of the complexity of the data, which you'll probably see in the presentation. So overall, the total number of zoos worldwide is quite difficult to quantify, but the majority of zoos will house species for ex situ conservation and breeding. And if successful with breeding, this means that individual animals might be born outside of their wild latitudinal range. And with latitudinal displacement comes changes in annual variability in photoperiod, and therefore changes to climate, seasonality, and annual mean temperatures. These are all drivers of physiological processes and therefore may affect reproductive success and survival, the very justification for them being in zoos. So we set aside to investigate whether weaning age, sexual maturity, or species average lifespans are affected in changes to photoperiod conditions because of latitudinal displacement, species-specific biological predictors, or individual life history characteristics. Additionally, we wanted to investigate whether the effect of changes in photoperiod conditions between the wild and the captive latitudes are modulated by species specialization. In other words, if an animal is classed as an ecological specialist, so has a low number of habitats and diet preferences, or a generalist, so a higher number of habitat preferences or diet preferences. So I wouldn't be here talking to you about this data if it didn't involve Species360 data. So obviously the majority of the information, the data that we got for this came from ZIMS. Thanks to all the wonderful keepers, record keepers that input all of these data. So it involved 162,138 individual carnivores for this, as you can see the complexity already. 22 species, and that was from 1900s to 2024. And from these data, we were able to pull and extract the birth and death date, the birth and average lifetime latitude, the number of moves these individuals had, and the species average lifespan. From the IUCN Red List, we were able to pull information about species or, where possible, subspecies range maps, and get from that the top and bottom of the wild latitude range and also the midpoint latitude. From various different data sets, so Panthera was one, we were able to get the age at weaning and the age at sexual maturity. And then we were able to find from ecological data, from home ranges to diet breadth, and parenting systems, social monogamy, and various other different elements that we thought might contribute as a potential variable. So from all of these data, we were able to pull out and create our own variables, which were survival to weaning, survival to sexual maturity, and survival to species average lifespan. And this was then looked at with regards to the annual photoperiod variation between the wild and the birth or average lifetime latitudes. Now, obviously, this is just a short overview of the data and how we've managed to get it. I could probably do about an hour's worth of presentation just talking about the data itself. So if you are interested in finding out more information about how we cleaned that data and what we did to it to be able to make it usable, then by all means scan the QR code and that will take you to our GitHub site with all of the information if you want to look at it. I can somehow no longer move there we go. In terms of the analysis, we did 3 Bayesian binomial phylogenetic mixed models. So this was according to each of the 3 different life stages that we looked at. Now, this is a summary of the model structure here, and obviously this slightly changed according to what we were specifically looking at. And some of those variables were not necessarily relevant to all of the models, but this is kind of the general general plan of what we did in terms of the modelling. So I'm not going to go into too much detail. I can do that later if need be. In terms of the results, I'll run through what these figures show. So on the y-axis, you can see the decades. So this is over time, how the success or the probability of weaning, the probability of survival to the various different stages occurred across the x-axis. So the first graph on here, this one here, is included all of the individuals after we cleaned all of the data, so 72,000 individuals. So as you can see here, the probability to survival of weaning has gradually increased over the decades. The next graph along is then of those the individuals that survived to a weaning age, how many of those then survived to sexual maturity? And the final graph is of those that survived to sexual maturity, how many of those then survived to the average lifespan age? So as you can see here, there's a very strong increase in predicted probability of survival to weaning and to sexual maturity between the early 20th century and the current day. And the graphs here are quite clear, but as you can see, the estimated probability of survival to weaning managed to reach 75% or 0.75 in the 2020s. So a massive increase over the years that we've been keeping animals or keeping records of animals. There was little impact of decade on the survival to species average lifespan, and this is likely because the impact of the management changes over time is largely concentrated on the increased survival in the early years. When we look at this with regards to the subspecies level, you can see that again, it's quite difficult. I've only included one of the graphs here, but as you can see why it's quite difficult to read that one. So 3 on a page would be even more difficult. But essentially you can see here that there's no species or family level clusters, and that variation according to the survival to weaning or to survival to reproductive age, etc., is definitely on a subspecies level rather than on phylogenetic. And this is shown here, I suppose, quite easily because there's quite a few Panthera pardus in terms of genus, but as you look at that, there's one just kind of second down from the top, which has got a really good success rate or probability to survival to weaning compared to the one very much at the bottom, which is Panthera pardus, which is much lower compared to its other subspecies. If we look at survival to weaning, there's a significant negative effect of photoperiod shift. So in other words, the more different photoperiod conditions are from the wild conditions, the lower the probability of survival to weaning is. However, this does change depending upon the species specialism. So a more generalist diet buffers this negative effect of the photoperiod shift. And there was no significant impact on social organization, mating systems, or parenting systems. So here, the biological variables or characteristics were not influenced or not influencing those changes. When we look at survival to sexual maturity and species average lifespan, there was for both milestones, there were no significant effects of the photoperiod shift, specialization indices, or the biological characteristics that we included. There was a very strong positive effect of the number of moves between institutions. And this surprised us a little bit because it was seen that the more that the animal moved from different locations, the more likely they were to survive to the next milestone. And we'll talk about that in a little bit. So significant positive effect of home range size as well. So essentially, the larger the home range of the animal, the higher the likelihood is that they were going to survive to the species average lifespan. So in conclusion, we can see that there was a dramatic increase in survival of zoo-housed carnivores over the last century. And we've put this down to, I suppose, the increased science and knowledge exchange to support the reproductive success of our carnivores within zoos. We saw that latitudinal displacements negatively affect survival to weaning, but this was not seen to other subsequent milestones. And it could be that survival to weaning age is the most sensitive of the life stages and therefore affected by physiological changes caused by the photoperiod. It's also quite useful to support further reproductive success of carnivores and potentially can also go to support breeding recommendations in the future. The individuals who move between institutions are more likely to reach sexual maturity and species average lifespan. So again, this was the one that kind of really throw us off a little bit because generally you kind of think that if animals move more, they're going to be potentially more stressed, which might put a more pressure on their reproductive success. But we think this is likely linked to the fact that generally only healthy animals who have a substantial health checks are moved for breeding. And also, if we know that the individuals are high on the breeding lists, as it were, then they might be given, their care or their health checks might be a little bit more intensive. And finally, the species traits such as social organization, mating, and parenting systems do not explain survival in zoo conditions. And these biological characteristics were not as important or relevant. And it might be that these are overruled within successful management techniques across zoos and breeding programs that we now run. So that's me. Thank you very much to Species360 for, of course, sharing the data, for all of those data inputters from all of the zoo members recording that data. And if anyone does have any additional questions or I don't get time to answer them all, please, by all means, feel free to contact me.
Samantha, what a great presentation and a great study. Thank you for that. I can only imagine with any successful project like this, how many more questions you have now that you have this insight.
Yes.
Yeah, so I'm sure others have questions for you too. So I'll open the floor for questions. Reke, what do we have?
Yeah, the first one is if the age of the mother is affecting the survival before weaning.
So we took the data as every individual. So we weren't looking at kind of the parentage and how they survived and how that then the kind of family tree elements. The main reason why we didn't do that was because the way that the data, I suppose, was we don't necessarily know that individuals are not moving out of ZIMS collections, and we also don't always know where they move. We'd have to then trace exact individuals where they're moving from and to. So we didn't look at it on a family tree basis. We looked at it as an individual basis.
Mm-hmm. So for the photoperiod, the effect only showed up at weaning, not at other life stages. So is that suggesting that the mechanism is more on the mother than on the offspring itself?
Yes, potentially. And again, it's, obviously from the very sort of factual data that we have from ZIMS, it's very difficult to be able to pinpoint exactly why. But I would think, something to do with the way that the seasons or the climate might impact on breeding seasons and things along those lines.
Mm-hmm. That's nice. Then we have one question. If you looked at zoos in northern versus southern climates.
We did. So one of the biggest challenges that we faced was that the majority of animals, and certainly carnivores, were moving from a southern hemisphere to a northern hemisphere. So generally, we know, that obviously we've got our global north as a predominant zoo, or is where a lot of the zoos are, and certainly a lot of the zoos are utilizing Species360 and ZIMS. So what we were finding was that there was majority of animals that were moving from the southern hemisphere to the northern, and I think we only had maybe even 1 or 2% of the data that was moving the other way around. So it was very kind of skewed towards the southern moving north compared to the north moving south.
Thank you. Then we have a question, if you can explain again that there was a difference in survival at the subspecies level.
Yes. So what we were thinking, or what we'd hoped, was that we could potentially look at this and look at how families, or even, genus level, would react in the same sort of similar way. But actually, from the figure that I showed, you can, well, hopefully see in the quick half a minute or whatever it was that I shared the graph, that it's actually not the case. And so the couple of species, I suppose, that I picked out was, Panthera pardus, and that was, scattered throughout all of that. And as we moved across, so as the images were moving from left to right, or as the curves were moving from left to right, that showed the change, or the increase in probability to survival. And so essentially what that was suggesting is that it's not anything that we can put down to in terms of the way that we manage leopards, for example. It's completely very subspecies relevant.
Thank you. I think that's what we can do for now. But I would just say it really surprised me as well that the transfers was increasing the chances of survival. But maybe we also put more effort on taking care of these individuals, because they are in general more important from the breeding perspective.
Yeah,
you.
that's the only thing that we could think of, because, like I said, you would think that more moves is going to be more problems, but it doesn't seem to be the case.
Which is also a nice result.
Exactly. Yeah. It just shows that, moving animals for breeding is not having a problem for their reproductive success. So positive
Absolutely.
overall.
Thank you, Samantha. There are a few other questions that you can answer in the Q&A.
Brilliant. Thank you ever so much.
Wonderful. Thank you much. And now I present our final speaker. Dr. Fiona Sach works at the Zoological Society of London, where she leads a global recovery initiative for species lost from the wild, and she co-chairs the IUCN Species Survival Commission's Extinct in the Wild Action Partnership. Over more than 14 years at ZSL, her career has spanned conservation science, elephant welfare strategy, and international partnerships. Grounded in a PhD in elephant ecology from the University of Nottingham, the expertise underpins today's talk, which draws on more than 60 years of ZIMS records to rethink what the data tells us about survival and lifespan in zoo-housed elephants. Fiona, off to you.
Thank you so much, Tim. Thank you for a lovely introduction. And thank you very much, everyone, for sticking with us this afternoon on this warm afternoon. So thank you. I'll just share my screen and we can get going. So we started working on this a couple of years ago, when we were really looking and thinking about how can we make sure we're getting better at caring for elephants. In the literature and in the media, there was a lot of articles and a lot of awareness being raised around compromised survivorship in zoo elephants. And this was something that we very passionately felt needed a bit of a examination and a bit of a consideration about, on a population level, what are the welfare trends for elephants. And we're in this amazing position to have decades and decades of longitudinal data on zoo elephants with Species360, held securely and safely with Species360. So we began collaborating together to really look at this and look at these population level trends in life expectancy and lifespan equality in our zoo elephants. So just thinking about the role of the elephant in the modern zoo. So a lot has changed since 1960 in how elephants are cared for in modern zoos. And a lot has changed in our care that we're able to provide, our public expectation, and the welfare needs of these animals as science has evolved. Elephants have really three principal roles in our zoos: to educate and to inspire people that hopefully will lead in time to conservation behaviour change, to garner and raise conservation funding and awareness, and in certain situations for conservation research, where sometimes in certain situations zoo elephants can support wild counterparts. Despite these very important roles and the funding which zoos have channeled into elephant conservation in this time period, elephants cannot be subjected to compromised survivorship and compromised welfare. And so it's very important that we use all the measures within our toolbox to assess welfare across the population and on an individual level. We have mechanisms, or we have some mechanisms in development around how we assess day-to-day welfare within our elephants in our zoos. But really, there's an absence of how this is looked at on a population level. And so this is why these measures of life expectancy and lifespan equality were used. We wanted the study to inform progressive zoos to think about how their evidence-based decisions around animal care can mean that elephants are able to live long and meaningful lives that are at least comparable to their wild counterparts when they're under human care. So I'm sure we've all seen in the media various reports and various publications that were highlighting the compromised survivorship of zoo elephants. And these were all taken from very real data, but from real data up to a point in time. And I think if we think about how elephants were cared for, really even up until the 90s and early 2000s, an awful lot has changed within zoos. But here we've got 4 of the main reports that highlighted this compromised survivorship and some of the quotes that have been used in the media. And certainly these were all very true. But we really wanted to look at the broadest and most far-fetched and wide-range data set. And we really wanted to think about how all the data could be included right up to current day so that we could have the best information available. There's lots of debate about the ethics of elephants in zoos, and there's proposed legislation across various countries around whether this is something that zoos should be continuing to do, and lots of information around perceived reduced survivorship. We very much felt that some of this analysis was outdated and really wanted to reflect the current day situation. And we wanted to make sure that any legislative change was being driven by sound evidence base. And we used an analysis of a very complete data set that really looked at proven longevity measures to reflect population welfare. So these measures, life expectancy and lifespan equality, these are very recognised measures of population welfare. And this example here is looking at humans, the human population in Sweden. And so life expectancy, so this is the theoretical average age of death. So this is the average age at which population dies. And lifespan equality, this is a unitless measure of the accumulation of death. Now, everything has to die, we must remember this, but what we want to have is a population that has a high life expectancy, so it lives for a long time, and that death is clustered together, so the lifespan equality is high, and so that concentration of death at older ages. And we see this in human society, and this is the example from Sweden from 1750 to sort of post-industrial society. And you can see that life expectancy increases, and the lifespan equality goes from being very spread out to being very sort of concentrated at the end of life. So why does this matter to our zoos? This is particularly with elephants, which are very long-lived. This is quite important because predicting the life expectancy of our animals can be very important in our long-term management planning to enable zoos to be able to provide for these animals. And it's really important to think about, the challenges that need to be overcome within young animals. So this example here is from the animals at Whipsnade. And you can see that these younger elephants have a shorter life expectancy because they do need to overcome those early life challenges that they face, particularly with elephants, things like maternal rejection, EHV, etc. So that was kind of why we were looking, and we looked and split it up between the 0 to 1, 0 to 10, and then the whole life of an elephant. So that's kind of why we made those decisions to choose those time periods. And we were looking at the Species360 data set. So this is the most comprehensive data set of elephants under human care. And it's about over 3,000 animals are included since 1960. And what was particularly interesting to us was the split between African and Asian elephants, and the split between elephants in their range state and not in their range state. So what we found was that we did have, a lot of elephants that were not a lot of elephants that were in their range state, that was species 360 elephants, for Asian elephants, but for African elephants, the absolute reverse. So most of our African elephants were out of range, whereas most of our Asian elephants were in range. So that's just something to bear in mind as we talk through some of the data and the results. But there's just something that we kind of looked at when we split the population up. We also had less data for male elephants. So particularly in the early time period, 1960 to 1992, we did lack data for male elephants. And that was just because there were less male elephants in the population at that time. We have to remember that even in Europe, we were still importing elephants from the wild in the early 1990s, and the sex ratio of import was massively skewed towards a higher female population. And so the data is just not, unfortunately, there for those male elephants. But I think that does highlight to us how much has changed around our philosophy of elephant care in this time frame. So when we look at the survivability, so this is the, we can see we have Asian elephants here on the right-hand side and African elephants on the left-hand side, females at the top, males at the bottom. And when we look at the survivability from the 1960 to 1992 versus the current period, so 1992 to 2024, we do see a really, really good increase in both our African and our Asian elephants. In our Asian elephants, we massively lack male data, so we do not have that data for the first time period. We don't have enough data. And then we were comparing this to studied wild counterparts. And what was particularly interesting to us as well was how little data there is in the wild on wild elephant life expectancy. So there were 2 studied populations for African elephants and 1 population for Asian elephants. And our Asian elephant population is a semi-wild population. So despite elephants being massively studied, there are still huge ecological knowledge gaps within the species. What was also interesting is that there is difference between the wild populations. So Amboseli and Samburu, we do see significant differences due to various factors, anthropogenic factors, climate change, etc., within those populations. And looking at lifespan equality, again, we do see progression in both African and Asian elephants from 1960 to current time. And we do see improvements today in our zoo elephants against our wild counterparts. But when we look at our out of range and in range, which we looked into a little bit more with our Asian elephants, we interestingly found that our in-range elephants are having a considerably better lifespan equality. So I think there's something there to think about in terms of how these animals are being looked after and climatic conditions perhaps that they're exposed to in range. So thinking about changes in how elephants are cared for, and again, here you can see a lot of these changes came about from the early 2000s, which is in the very, very recent past for an animal that lives so long. There's been phenomenal change in how elephants are looked after. And from looking at this, we realized over the last 20 years, every other year, there has been a new best practice guideline or a new legislation around how we care for elephants through EAZA, or AZA, or BIAZA. So you can see that this progression has really come in recent times. For example, sand flooring, this really came about in 2003, and now, really only today, are we seeing those improvements coming through in foot health. So this slide here just highlights, in very brief summary, lots of this data, there's lots of numbers on here, but it's all in a publication that's just come out in Scientific Reports. So please take some time to have a look at it if you're interested. But we really hope that this work will inform progressive zoos into making evidence-based choices so that elephants can live long and happy lives under human care that are at least comparable to their wild counterparts. Specifically like to thank Species360 for working with us on this, and our collaborators at the Max Planck Institute and the University of Southern Denmark, and every Species360 member that has contributed this data. And to circle back to the point made at the very beginning, it all starts with a single data point. None of this would be possible. Looking into any of this on a population level would not be possible without all of the contributions of our Species360 member zoos. So thank you.
Thank you, Fiona. It has really been a joy to see this research come together, and thank you for your close partnership in doing that. And of course, for ZSL's ongoing support for the Conservation Science Alliance as well. Nanette, what questions do we have for Dr. Sage?
Yes, this person said it's fantastic to see this good news story. And in their original work, it was speculated that psychological stress and being overweight could be independent contributors to shortened lifespans. Any idea what has improved? Their slimness, their happiness, both, or is it all about foot health?
That's a great question. I think the way that we feed elephants has massively changed in really the last 10 to 15 years, and moving away from these very high sugar, high fruit diets that do have a propensity towards obesity in our elephants. I think our awareness around obesity in elephants has also massively increased. And some of these reports have really shone a good light on that and forced zoos to think more carefully about what their elephants are eating, how much their elephants are moving, and how much they're being fed under human care, which I think has massively helped in terms of reducing that obesity. And this clear link between obesity and reproductive output in our female elephants, and really making sure that we are maintaining appropriate body conditions for these elephants, particularly these young elephants being born today under human care, making sure they're not moving into that obese category.
Do we know the proportion of maternal rejection out of known breeding events in zoos or in the wild? What are the reasons for maternal rejection?
Gosh, that's a big question for 5-minute answer. I think it's very multifactorial. Hugely depends upon the elephant, social structure of the elephant, the welfare and well-being of that individual at that given point in time, probably considering the birth that animal has experienced. So I think there's a lot of key factors to be considering around maternal rejection. I think what we found in our publication, and it's nicely articulated in one of the figures, is that that 0 to 1 window in the wild is really difficult to monitor elephant death. Finding baby dead elephants is really, really challenging in the wild. So we do have good data for that, particularly in the more recent time period in zoos. I think going back in time when ZIMS was, well, before ZIMS, but when records were kept in a very different way, it was probably something, again, that was recorded with less meticulous care than it is today. So I think some of this data does have to be taken with a pinch of salt.
And this might be the last question. Can you talk more about the differences noted between African and Asian population life expectancy, the male African versus male Asian? Why do you think it's a greater percent for African males?
Under human care or in the wild?
It's not specified.
Okay. So I think we must also remember that these populations if we're talking about the wild, these are very specific populations, and data is collected obviously to the best of ability. But I think, there are limitations to how you collect wild data on life expectancy. And so I think that might come into some of the changes that you're seeing. And the differences in the populations, those two wild populations experience quite significant differences in poaching and other anthropogenic activities, as well as climate change and drought, which may explain some of the differences that we do see, particularly some of the shock, sort of dramatic shocks in the data.
Thank you.
Yeah, great. Thank you very much. So that brings us to the end of our presentations, and I'd like to take a moment to reflect on what we've heard. 6 talks, 6 very different species, from geckos and squirrels to echidnas, carnivores, and elephants. On the surface, they have little in common, but look closer and the same thread runs through every study. Shared data brought together across institutions, answering questions no single zoo or aquarium could answer alone. We've seen that data inform animal care. Decades of records can rewrite what we thought we knew about survival and lifespan. Clinical and post-release data can guide husbandry and conservation for cryptic species that are notoriously difficult to manage and study. And detailed breeding records can reveal what drives reproductive success and where more consistent record keeping would help. We've seen shared data inform conservation. Understanding how latitude and photoperiodic conditions shape the lives of zoo-housed carnivores helps us plan for and provide high standards of welfare for these species. Filling the gaps in what we know about understudied geckos gives breeding programs the evidence they need and what they've been missing. The better data on threatened species held in human care directly supports the breeding and reintroduction work at the heart of the One Plan approach. And we've seen it inform institutional species planning. Aggregated data lets us ask direct questions of our whole sector. How do we prioritize which species are in our care? Are we housing the species that most need our help? Those that tell a great conservation story? Or species that bring visitors to our institution, driving the revenue that supports our work? That is exactly the kind of questions this data allows us to ask and to answer honestly. None of this happens in isolation. Every insight you've heard today began with a keeper or veterinarian entering a record, a member institution choosing to share its data, and researchers willing to work across borders and disciplines. That is a conservation science alliance at its best. I want to end by saying thank you. Thank you to our speakers. Thank you to the members whose data makes this research possible. I also want to thank one more time our current CSA sponsors for their support. If you or your organization would like to learn more about supporting the CSA, you can scan this QR code. And finally, thank you to everyone who joined us today, whether as a presenter, a sponsor, or an attendee. This community is what makes this network and this symposium possible. I hope to see you all again next year. We cannot wait to celebrate another year of impactful science, helping wildlife thrive. Take care.